Alright students, today we're going to be learning about the cell membrane and all the structures that it has. Let's take a quick tour of the cell membrane and how it maintains homeostasis. Remember, the cell membrane really has four functions. It really wants to control what goes in and out. We don't want to let everything in, so we have this barrier. This is called a phospholipid bilayer. Phospholipid because that means two layers. Each one of these looks like a little sperm and two tails, but they're not related. They're not even cells. These are molecules. Again, here's the glycerol portion, and here's the fatty acids. Two fatty acids. All combined, this is one phospholipid. So phospholipid has a glycerol and two fatty acid tails. The glycerol or head portion loves the water. So inside is the water. Also, we have water on the outside, so they love to face the water. Inside, these are hydrophobic. They do not like water, so the tails face each other. And again, these are hydrophilic. The heads love the water. So this forms our barrier. Remember, it's not fully permeable. If it were fully permeable, everything would go in and out. If it were non-permeable, then nothing could get in, and then the cell would starve. The reality is it is semi-permeable. And remember the key thing for things to go in. They must be small and uncharged for them to go through the phospholipid bilayer. And things that are bigger, or they could be small but they're charged, they won't be able to enter. And that's how the cell is picky about what enters. We don't just want everything to go in. We want things like bacteria, viruses to stay out. We want certain substances to not be able to enter. Only the things that it needs to enter. Sometimes there's things that need some help to get in, and so we'll talk about those. But for the most part, it's a barrier that prevents some things from entering, but allows others to enter. That's called semi-permeable. So that's our phospholipid bilayer. And a couple things, let's talk about, there's two types of ways to get things in. We can either use active transport or passive transport. Let's start passive transport. So I'm going to talk about the four that are passive, meaning they don't require energy, and it all relies on diffusion for these four. Diffusion is the movement of substances from high concentration to an area of low concentration. Let's start with the basic one. So I'm going to show carbon dioxide right here, and that's our CO2. And carbon dioxide is small enough that it can wiggle through and go in and out without a problem. It goes in very easily, and because of that we call this simple diffusion. Diffusion that was simple, that was easy because it was small and it was uncharged. Simple diffusion. The same thing can happen with water and oxygen, which I don't have shown here, but I got another carbon dioxide molecule. Cool. So it goes from high to low, from where there's a lot to where there's less. Another structure or another process has to do with osmosis, and that's when we have some water. Again, this is all diffusion, so from an area of high concentration to an area of low concentration. So for example, there's 3 water molecules out here, 1 water molecule in here. It wants to go from where it's a lot of it to where it's less. So it'll go from here to here until there's what's called equilibrium reached. And again, it was like simple diffusion, but because it has to do with water, it gets a special name, and we call it osmosis. I'm going to move this to the left a little bit. So far, keep in mind what we have. Simple diffusion, we have osmosis, which is also simple diffusion, but of water across the cell membrane. And then the next thing, sometimes we have some molecules that are very big and they can't go in. So something like glucose, maybe an amino acid, and they can't go in, and because they're too big, so it'll just bounce off. It can't get in and needs some help. So we have some carrier proteins that do something really cool. These carrier proteins will allow it to come in, and again, so now the glucose can enter, and once it enters, it kind of changes the shape a little bit and kind of spits it into the other side. And let's say glucose needs to exit. Maybe we have too much or we need to get rid of it. It'll go up through here and then we'll spit it back out. So this is a really cool little structure that we have. Again, this made it easier for glucose to enter, and in Spanish, easy means fácil, so think of it as facilitated diffusion, to make it fácil or easy for big things to get in. And again, remember it changing shape. So that's our third one so far. Now, sometimes there are, found our oxygen. There goes. Let's take oxygen back to simple diffusion again. Now, then we have some molecules, or not molecules, but ions. They're small, they're charged like sodium, it could be potassium, it could be iron, anything with a plus or negative charge. And these are just going to be bouncing around all day. They can't enter. They're positively charged or negatively charged, and they repel, kind of like opposing or similar magnets, right? And they can't get in. But fortunately, we have another type of carrier protein. And this carrier protein is called an ion channel. And this will allow it to go in, or come out. So we call it an ion channel. And all these were, again, passive transport because it had to do with molecules or substances going from high to low concentration. And just think about it. When you're going from high to low, from where there's a lot to where there's less, it's like going downhill on a bike. When you go downhill, you don't need a pedal. You don't need energy. So it's automatic. And so it's like passive transport, meaning no energy required. That's diffusion, high to low. Okay, so now we talked about passive transport. Sometimes we need energy to get things in. So let's go to the right side now. So let's say we have a big molecule, and let's say, and I'm talking big, I don't mean like amino acids, I'm talking about not monomers but polymers. So let's say this is a big protein, and this protein needs to exit the cell. And so it'll have a piece of the membrane, which I don't have here drawn very well, but pretend this has phospholipids all around, and this will be what's called a vesicle, and the vesicle will come up to the surface and it will actually merge with the cell membrane and spit this out, and we get something big out. That's exocytosis. Think exo, exiting. And sometimes we need to get something big in, so the same thing will happen. Something big will enter, and then it'll form a vesicle with a piece of the cell membrane, and that'll come in. I'll show you an animation of that later. And that is called endocytosis because we're allowing something big to enter. And then we have active transport, or another part of active transport, because this was also active transport. And remember that even this needs ATP energy to happen because we're taking something big out from lower concentration to higher concentration. So in this case we have ions that need to be pumped from an area where there's a lot, or from where there's a little bit to an area where there's a lot. So we'll use, again, ATP. This time we're going backwards. We're going from low concentration to high concentration. So think about riding uphill. You need energy when you ride uphill from low to high concentration, because you're going from an area that's not as packed to an area that's fully concentrated. It takes a lot of energy. It's kind of, think of like going on a bus, and it's a packed bus. There's a lot of people on it already, and so if you get onto a packed bus, it takes a lot of energy to find your seat because you're pushing against all these people in a crowded area already. So think about it that way. That's why it takes energy. Okay, so we have three examples: endocytosis, exocytosis, and active transport. Or sorry, protein pumps, which are all forms of active transport. Before I forget, here we have some cholesterol molecules. And cholesterol molecules just hold the membrane together. It keeps falling down. There we go. And without these, the cell membrane would collapse, all the water would leak, and so the cell membrane keeps this very sturdy and fluid at the same time. They call it the fluid mosaic model, which just means that the cell membrane is constantly in motion, moving. These proteins can constantly go back and forth, so they're floating around, and that's actually advantageous so that in case there's molecules over here that need to enter, we're able to go to where the molecules are at. So they're constantly moving back and forth. All these proteins are kind of like bouncing around in the liquid, kind of like a rubber ducky floating around in your bathtub. So again, we have a cell membrane which maintains homeostasis, controlling what goes in and out. We have the phospholipid bilayer. We have four types of methods of four types of passive transport which require no energy: simple diffusion, osmosis, facilitated diffusion, and ion channels. And then in active transport, we have three types: endocytosis, getting something big in; exocytosis, getting that big thing out; and protein pumps, or ion pumps, where we pump ions from low to high concentration. And that's pretty much it.